Hi everybody, in this video we are looking at the process of selective reabsorption. So to put it in context, we've already learned that ultrafiltration happens here in the Bowman's capsule. So substances from the glomerulus, from these capillaries, are filtered through, and that's the first stage there of the whole nephron. Once the filtrate has been formed, it travels through the proximal convoluted tubule, and this is where selective reabsorption happens. If we just then follow it through from there, after selective reabsorption, then we move through the loop of Henle, and then we move up through the distal convoluted tubule, and then finally down to the collecting duct, and the urine is what we end up with at the end. This part here, as we said, is what we're looking at today, so the selective reabsorption happens in the proximal convoluted tubule. Another thing which is important is to remember that surrounding this whole nephron we have this network of capillaries. This is a simplified version, and when we look at diagrams we'll make it even more simplified, but that very close contact with capillaries is really important for the processes that we're going to look at. Okay, so if we start from here then, and what we want to know first of all is what's the purpose of selective reabsorption? Why does it have to happen? If we start off with our proximal convoluted tubule, so this is our tubule here, so here is one of the walls of the tubule, and here is the other wall, which means that in the middle here, this is where we'll find the filtrate. So this filtrate has moved down from the Bowman's capsule, and it will now continue through the proximal convoluted tubule. In this filtrate are various substances, things like glucose, salts, amino acids, and the body does not want to lose all of these to the urine. So the purpose of selective reabsorption is to take these substances which are in the filtrate, which were filtered through during ultrafiltration, and move them back into the blood. It's called selective because at this stage, unlike what happened in ultrafiltration, where in ultrafiltration substances would just move through if they were small enough, this process here is selective because the body is able to control which of these substances and how much of these substances moves through. So for example, all of the glucose gets reabsorbed. You don't end up with any of the glucose continuing through and ending up in the urine. Depending on the levels in the blood and the body's needs, then the body would reabsorb some salt, some amino acids. So if that's what selective reabsorption does, how does it do it? If we look at our structure again, but we're going to look at it in a bit more detail this time. So here's our filtrate, and then here's our blood capillary. Now, at the moment, I haven't drawn any cells in the walls. So here's our capillary wall. This would be made up of lots of epithelial cells, and the same here, and then the same for our proximal convoluted tubule. What I am going to do is redraw this wall here. So this wall of our proximal convoluted tubule is important because this is where the process of selective reabsorption takes place. So if we look at these cells, they have a particular shape and they have a particular structure which is very important for the process. We've also got a couple of things to point out. So this purple part is the basement membrane, a bit like we had and we saw in our in between our glomerulus and our Bowman's capsule. So the basement membrane is not part of the capillary, it's actually not part of the proximal convoluted tubule either. It's in between the two and it acts as a barrier. Not to be confused with the bottom lining, like the bottom part of our proximal convoluted cells, which we call the basal membrane. So this cell here is the cell of our proximal convoluted tubule. It has an edge here, which is next to the filtrate, and then it has an edge here, which is nearest the capillary. The side of the proximal convoluted cell that is nearest the capillary is the basal membrane. So this whole layer here is our basal membrane. And what you can see is that the basal membrane is folded. It's folded because it gives it a larger surface area. We can also see that on the surface that's closest to the filtrate, it's actually got microvilli, again massively increasing the surface area. And the other thing to notice, if you look in between those cells of the proximal convoluted tubule, you can see that they look quite thick. So we have these very tight junctions. And that means that substances, we've already said that substances are going to move from the filtrate into the blood. And these tight junctions mean that those substances have to pass through the middle of the cells, where the cell can control the movement. It can't just pass through in between the cells. Okay, so we're going to just zoom in a little bit so we can see what's happening in these proximal convoluted cells a little bit more detail. So I'm going to just use a G, so G for glucose, but this represents all of those solutes. So the same thing that will happen for the glucose will happen for a salt for our amino acids. So I'm just simplifying it by using a G to represent glucose and therefore all of the other solutes that will get reabsorbed back into the capillary. Now the first stage of this process happens in the basal membrane, and what we've got here is a sodium-potassium pump. So this sodium-potassium pump in the basal membrane, and there will be lots of them all along here, and that's partly why we need a big surface area, because it means we can have more of these sodium-potassium pumps. The job of this is to pump sodium from inside the cell out into the capillary. So this is pumping, it's active transport, it's using energy, and there will be lots of mitochondria as well in this PCT cell to provide the ATP for this transport. It's a sodium-potassium pump, which means that potassium comes back inside, but we're going to ignore that at the moment because we don't need to focus on that. We're just interested in the sodium. Okay, so sodium is pumped out. What that means then is that the concentration of sodium inside the cell is going to decrease. We know that we've also got lots of sodium in our filtrate. So because sodium has moved out into the capillary by active transport, and that has reduced the concentration of sodium inside the cell, that means we now have a concentration gradient. We have a higher concentration in our filtrate and a lower concentration in our PCT cell. So what this is here, this is a co-transporter. So this is a protein in the membrane, and again there will be lots of them all the way along here, all along the microvilli. This co-transporter transports sodium passively, so it's just going down its concentration gradient. Because it's a co-transporter, that means that it also takes something else with it. So this co-transporter takes glucose at the same time as the sodium goes through. This means that the glucose, which actually needs energy to move across, is being moved actively, but the important thing is that no ATP is needed for this to take place. This co-transporter is able to use the movement of the sodium to drive the movement of the glucose. So it's an active process, but it's not using ATP. We call it secondary active transport, because the only reason that this can happen is because this sodium-potassium pump set up the concentration gradient in the first place, and this sodium-potassium pump needed energy, ATP, to do that. So, secondary active transport moves glucose along with sodium ions into the cell. So now we've got, again, concentration gradient. So you see that those substances, the solutes, the sodium and other solutes inside, the glucose and other solutes, they're going to move down through the basal membrane, but this time just move by diffusion into our capillary. So I'm just going to remove all of those now just to make it a bit clearer. So what we now can see is that if we've now removed lots of solute from the filtrate, the water potential is going to increase. Those solutes have now moved across into the capillary, which means the water potential decreases. So now we've got a water potential gradient. So the next thing that's going to happen is that water molecules from the filtrate are going to move down their water potential gradient into the PCT cell and then into the capillary. And that's the final stage of selective reabsorption. So we started off with the movement of sodium, and then we've got the movement of glucose and the other solutes, and finally we've got the movement of water. Okay, that's all a reasonably complex process, but hopefully that makes it clear. Thank you very much.